Good afternoon, everyone. I'm Colin O'Connor, Custom Content Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Developing a Miniaturized DNA-seq Method to Reduce Costs with a Next-Generation Liquid Handling Platform, and it's sponsored by SPT Labtech. Our speaker is Thomas Brabbs, Senior Research Assistant in the Genomics Pipelines Group at the Earlham Institute.
Is sort of like divided into three parts. So the first is sort of like the utility or the use of low-cost library construction and methods for genomic studies. Next is an overview of the protocol that we've developed with SPT for their Firefly platform, and then the final one is a validation experiment that we performed to compare the miniaturized libraries versus a full-size library. So before I start, just a quick introduction of what the Earlham Institute is. So we're one of the institutes in Norwich that form part of the Norwich Research Park. So we're the one circled on the slide. The park also includes the University East Anglia, the John Innes Centre, the TSL Quadram Institute, and the Norfolk and Norwich University Hospital. So what does Earlham itself do? So we are a genomics institute, so we focus on bioinformatics, genomics, technology development, and synthetic biology to answer like urgent global challenges, so food security, biodiversity, etc. To facilitate this, we have some core platforms that we can utilize. So there's the Genomics Pipeline Group, which is what I'm part of. So this is a genomics sequencing team, and then a single cell group and a core bioinformatics group in there. There's the BioFoundry for synthetic biology, the research e-infrastructure, this is like high performance computing, and then we have advanced training as well for bioinformatics and genomics techniques. So moving first to the first part of my talk, which is the utility of low-cost library constructions. So firstly, sequencing costs. So I'm using here Illumina and the benchtop sequencers as the example. So you can see going from the top, we have the MySeq, then the NextSeq 550, and then finally the most recent NextSeqs, the 1000, the 2000. And then underneath each, I've put how many paired end reads, or read pairs, should I say, that they produce. So you can see from that their numbers have increased east to the newer one, which means that you can put more samples onto each sequencing run. So the consequence of this is that the cost per sample has decreased east dramatically. So there should also say that this is also true of their production machines. So the HiSeq 4000 versus the NextSeq, you have a halving of the time per run and a doubling of the yield per run. So the advantage of this cost reduction sample is obviously you can now have larger studies with more individuals. So this could improve your statistical significance if you're doing RNA-seq by having more biological replicates, allows you to look at rare variants, and then enables you to do bigger population studies. The other part is that you can democratize genomics. So you're bringing the costs down of doing a genomics experiment, allowing more people access. So although the cost of sequencing has come down, we need to also consider the sort of like upstream costs of that as well. So what I mean by this is collecting the samples, then extracting DNA or RNA from the samples, then library construction, and then to the sequencing. So the sample collection, extraction, and library construction can all add up to then bring the cost per sample up again. And so for this, I'm only going to focus on the library construction and part. So in terms of the cost for library construction, there's four main parts to it. So the first one is labor. So the number of hours that a person has to work on the library construction. Then you have reagent costs, so the cost of the enzymes, the primers, etc. Then you have labware, so the cost of tips, the cost of plates, tubes, etc. And then finally you have the cost of the equipment, so the PCR machine, the pipettes, etch, the QC equipment. And so for this, I'm going to be mostly focusing on the labor and the reagent costs. So equipment tends to be quite fixed for that lab in terms of the costs. Labware, you can reduce costs, but by comparison to labor and reagents, the cost reduction isn't quite as significant. So in terms of reducing the costs of the labor and reagent costs, so there's two things that can be done here. The first is miniaturization. So this is for reagent costs. So the reagents themselves are fairly fixed in their price from the manufacturer. So the only way to reduce costs is then to use less reagents in terms of volumes per sample. So then miniaturization, the library preparation will allow you to do that. The second is automation. So this will help reduce the labor costs as you then have the robots to do more of the pipetting in a library preparation steps, allowing you to have less hours worked. So in this regard, at Earlham, we've developed a low input library construction method. So this is based on the Illumina Nextera chemistry. So this was first published in the paper on the right-hand side of the slide. So this was developed by Darren Heavens in the genomics pipeline group. So it's achieved a 1 in 10 miniaturization of the reagent volumes, and then we're using a 1 nanogram input into library construction. So from this miniaturization and the DNA input, we've been able to reduce down the reagent cost to $10 a sample. And then as a proof of concept of this paper that I've inked in the slide was able to sequence 10,000 salmonella genome samples. So this protocol is low input transposase enabled, which I'll refer to as light for the rest of this talk. So I'll just go through the light workflow with you. So when DNA samples arrive in our lab, first we do DNA QC to measure their concentration. So this is using a plate reader and then the mosquito to set up. We then use the mosquito robot and the dragonfly robot to normalize the DNA down to the 1 nanogram input. But from that, we then go into the tagmentation reaction. For this, we use a Fluent 780 and a off-deck PCR machine. The same aim are used for the following PCR amplification reaction, and after which we then go to a B cleanup. This is done on another 780. From this, we then check the libraries for quality control purposes. So we use a mosquito and a GX touch. So this is to look at the library size and the library concentration. From this, we can then pull the libraries to a equimolary, and then the final step we use is a size selection using a pippin, a blue pippin. And so for this, we are selecting for a narrow band of DNA, of library fragments, because then this is optimized for 150 base pair paired end reads so give like the optimum and sequencing results, rather than having a wider, not size selection, having a wide array of fragment sizes. And then the final, after it's done size selection, is obviously sequencing. So in terms of the light pipelines usage, so we're averaging between 10 to 15,000 samples per year, and then this consists of 125 projects on a wide range of species. So I've given some examples here. So we have Trinidadian guppies on the left, and then Arctic arthropods, Shigella on the right, and then various UK wild plant species. So the one I've shown here is Herb Robert. So from these projects, they also showed another advantage of low input library preparation and techniques, which is the fact that they are low input. So the advantage for this is that for samples where you do not have much DNA, so in the example here, I have a collection of beetles. So the one circled in blue, you would expect to yield enough DNA for a standard library preparation technique, where you need hundreds of nanograms of material. However, for the one in red, you might not be able to get enough DNA from it. So for this one, you could use a miniaturized low input technique and then still be able to get a genome sequence out of it. So just in conclusion for this part on the utility of low-cost library construction techniques. So it allows for larger studies and then lowers the barrier of entry. To reduce costs, miniaturization and automation are key to this. And the other added bonus of low-input miniaturized protocols is you can then sequence low-yielding samples. And then the final one is that light shows the advantage in viability of these methods. So then I'm now going to move on to the miniaturized Illumina DNA Prep protocol. So firstly, why did we choose the Illumina DNA Prep to miniaturize? So we were wanting to update the light pipeline as the original Nextera is coming to the end of its life. We're looking at different chemistries. So for the DNA prep, this is the latest version of the Nextera chemistry from Illumina. So its key difference to the others is that the transposases that add the adapters to the DNA are now bound to a bead rather than free-floated in the liquid. So the advantage of this is that first it normalizes for DNA input. So above a certain DNA input, you'll still get consistent yield and you'll also get consistent library sizes. Illumina advertises it as having improved low and high GC tolerance. And so it's more capable of having reasonable coverage at the low end and then at the high end. And then finally, the volumes of the DNA library prep technique is amenable for miniaturization. By that I mean that when you reduce the volumes, none of them fall below a point that a robot or a human would be unable to pipette them accurately. So for the miniaturized DNA prep technique. So for this, we decided on 5x miniaturization. So this is the sweet spot between reducing costs, but also keeping the volumes within a pipettable range. And then we're using a 384 plate format for this library prep method. So this helps reduce labware costs because you're only using one plate for 384 samples. And the other thing to note is that we made some volume adjustments for various reasons. So these include like going over a maximum well volume, improving pipetting accuracy, and preventing bead carryover, which I'll come to when I go through the workflow for this miniaturized effort. So because of our use of the dragonfly and the mosquito in part of light, and also for our single cell library preparation techniques, we decided to partner with SPT Labtech to develop this miniaturized protocol, and then we developed this for their new Firefly platform. So before I get into the workflow, I'm just going to go over what the Firefly is with you. So the Firefly is a new liquid handling platform from SPT Labtech. So it has a small form factor, and the reasons for choosing this one is that it has a dispensing head, which is based on the Dragonfly, so it's very accurate at small or volume dispenses, which makes it ideal for a miniaturized platform. And it also has a pipetting head, which is capable of 384 or 96. Loading 384 or 96 tips, so you can do full plate transfers or partial plate transfers at once. Plus, it also has a shaker and cooling in points for a plate and the reagents. So it makes it allows us to perform the DNA prep method on it. So I'm just going to show you some videos of the Firefly in action. So the first one I'm going to show you is of the dispensing head arraying into a plate. So this is the Dragonfly equivalent. And so this should start shortly. So first the deck moves out of the way, and then the dispensing head, which is on the right, will then move down to the liquid. I should add that the liquid in this case is colored to enable it to be more or visual during a video. So at the moment it's doing a pre-wet to wet the syringes so that it can aspirate and dispense accurately, and now it's aspirating the liquid for the dispense, and it then comes back out, and then it's going to dispense into the plate on the right on the top of deck. So it's going to do this in a row-wise manner, but you can also do it in column. And then, so for this, I'm only going to show you it fill in half a plate, 8 for this. It takes about 20 seconds to fill the plate. So the next video I'm going to show you is of the pipetting head and also the gripper arm, which is also part of that head. So for this one, the pipetting head will aspirate from the plate on a magnet. So this is simulating a bead cleanup. So it's going to aspirate the supernatant off the beads and then transfer it into a waste plate. 8, and then once it has finished dispensing, and then it will then perform a tip touch to ensure that there is no liquid left on the tips. Then once it comes out of the plate, it's then going to eject the tips back to their original position. And so the tips are held by a metal clamp that allows it to be unloaded and loaded onto the pipetting head. And then finally, the last thing I'm going to show is the grippers as moving the plate off the magnet it onto a normal deck position. So, now moving to the actual workflow that we developed for the miniaturized DNA prep workflow. So, for the tagmentation, we're using the dispensing head of the Firefly to array the tagmentation reagents into a 384 plate. We're then using the pipetting head to transfer DNA from a source plate into the 384 plate, and then the pipetting head pipette mixes the DNA with the tagmentation reagents. This is then taken off deck to a thermocycler for the tagmentation reaction. From this one, it then goes into the stop tagmentation reaction. So for this, is we're dispensing in the tagmentation stop buffer, and then for this we're actually using the shaker to mix the stop buffer with the tagmented DNA, and then this goes back onto the PCR, onto the thermocycle, sorry, for the stop reaction. After this, we then go into the tagmentation cleanup. So for this, the plate gets put onto a magnet on deck. The supernatant for this is then removed to a waste plate, and then the tagmentation wasp buffer is then arrayed into the plate. I should add for this that the wash buffer volume has been reduced. This is related to preventing spills when we resuspend the beads in the wash buffer via shaking. So the beads are then resuspended by shaking, and then this goes back onto the magnet, and this whole cycle is repeated 3 times. So after the final shaking, it then goes on to the magnet. It's for a 4th time, of which the supernatant is again removed, and then PCR reagents are then added, and then shaking is used to resuspend the beads in the PCR master mix. So for this, we've reduced the PCR master mix its volume slightly, which I'll come on to the reason why for the next one, which is we then transfer in the primers and then mix the primers with the master mix and the beads. So we've increased the volume of the primers to 4 microliters to improve pipetting accuracy at this point. So we achieve this by removing some of the water from the PCR master mix and then transferring it into the for the primers. After this, they then go into a 5-cycle PCR on the off-deck thermocycler, and then we move into site selection. So for this, there are 2 things that happen concurrently of each other. So firstly, another 384 has beads and water arrayed into it. At the same time, the original plate is placed on a magnet, and then the supernatant on that is then transferred into the new bead plate. It's mixed, it's incubated for a while, at which point we then repeat the cycle for the second spray, of which another bead plate is set up. The original bead plate is placed on the magnet, and then the supernatant is then transferred to this new bead plate, mixed and then incubated. After that, we then place the plate on a magnet, remove the supernatant to waste, and then we begin the ethanol washes. So for the ethanol washes, these have been reduced. This is to prevent an overflow of the 384 plate, because the miniaturized volume would have overflowed the well to a 384 plate. So for this, we dispense in the ethanol, incubate, and then remove the ethanol, and then this is repeated 2 times, all while the plate is still on the magnet. After this, we then go into elution. So for this, we have increased the amount of resuspension buffer that we're using in here. This is to prevent bead carryover for the final elution. So this is dispensed into the plate 8. The beads are then resuspended by pipetting and incubated, and then the final elution is the plate is then put back onto the magnet, and the eluted libraries are then transferred off to a new clean plate. So just to conclude this section, and so we have developed a 5 times miniaturized DNA prep method. So this is using a 1/5 of the volume of the standard DNA prep method, and this is in a 384 format. And we've utilizing the functionality and capability of the firefly to carry out this library prep method. So now move on to the validation experiment we performed to test the miniaturized protocol. So just go for an overview of the validation experiment. So this is using 2 control DNA samples, of which I'll come to on the next slide, and then with 3 different library preparation types. We have the 5x miniaturized that we developed. So we have a full volume manual DNA prep method to compare it against, and then also we have our light method. Then all of these libraries were sequenced on a MiSeq using 150 base pair paired end reads. And the main thing that we're looking at for today's webinar is the low and high GC tolerance. So going into the DNA samples in a bit more detail. So the two DNA samples are human, which is a mix of male and female DNA, and then a microbial mock community. So in the table that I'm showing on the slide, you see all of the species that are in this mock community. So there's 22 in total. And then you can see in the middle column you can see they have varying GC percentage, of which the graph I've got on the right shows the GC percentage plotted. So you can see that there is a group that have high GC and there's a group that have low GC. So for this talk, I'm going to only highlight 3 of the species from this group of 22. So those are Clostridium benjerinki, which is the lowest GC content at 29.2, so it's 80 rich. Escherichia coli, which is median and GC content of 50.6, and then finally Rhodobacter spirophores, which is 68.8, so it's GC rich. Then a little bit more detail on the libraries. So firstly, we have the 5x miniaturized DNA prep libraries. So we had 24 for the human and 18 for the mock community. So for the human, this is then subdivided into 6 replicates using 100 nanograms, 20 nanograms, 6 nanograms, and 1.2 nanograms. For the mock community, we had 6 replicates of the 20 nanograms, 6 nanograms, and then the 1.2 nanograms. So the reason behind using the different DNA inputs for these libraries is to test out the bead normalization and to see whether the bead normalization is still working when we miniaturize. So for the 100 and the 20 nanogram inputs, these should still be assuming in that the miniaturization and normalization reduce at the same aim rate. These should equate to the recommended input range from Illumina of 100 to 500 nanograms for the full volume kit. Then the 6 and the 1.2, the reason for having those is to then test more of the low input library preparation methods, similarly to light, to check whether we can have a similarly low input to enable all this methods used for samples with a lower DNA yield. And then finally, for the full volume, we have 3 human and 3 mock community, all of which are using 500 nanogram input. And then for light, we have 6 human and 6 mock, and these are all using 1 nanogram input. So first I'm going to have a look at the bead normalization. So first I'm showing here is a table for yield. So on the y-axis you have yield in nanograms per microliter, and then on the x-axis we have the average for the different DNA and samples and DNA inputs. So I'm not showing light here, I'm just showing the full volume and the miniaturized DNA prep protocol. So from what you can see from this is that, as you can see, that as the input decreases, you're getting also a decrease in the yield as well. So this would tell us that the yield normalization is not working when you miniaturized. The other thing for normalization is that it also normalizes for insert size. So this, in contrast to the yield, is actually still working. So as you increase the input, you do not get an increase or decrease in the insert size. For the lowest inputs, there is a small decrease in size. However, this has been reported by Illumina themselves for very low inputs, that you tend to get a slight drop in the insert size. So this is to be expected. So for the analysis to compare the GC content, so I'm just going to go into the analysis bioinformatic pipeline in some detail. So firstly, for human, we have the human samples coming in. They're then mapped to the human reference using Bowtie2. The resulting BAM files are then used as input into Picard tools, collect GC bias metrics, which then outputs the GC plots that we're using to compare the different library preparation techniques. So for the mock community, for this, we have created a single combined reference of all 29 species into one. So the reason for doing this is for a read, if we were to do each 22 species individually, it could then map to multiple species. Whereas if we have them all in a single combined reference, it will map to the one that is the best fit for that particular read. So this will remove any chance of bias that we're creating from having reads that map to multiple species. So for the microbial mock community, the actual pipeline itself, after creating this reference, is you have the mock samples being mapped by Bowtie using this combined mock reference. And however, this we then filter the BAM files for the individual species to create a BAM file for each individual species for that library, and then this is used as input for the Picard tools collect GC bias metrics script. So here I'm showing an example GC plot from the Picard tools, also just to explain what it is actually showing. So on the x-axis, we have GC percentage of 100 base windows. So the way that this is working is that it divides the genome into 100 base segments, and it's called windows, and then measures the GC percentage of that window. So on the bottom here in red, you can see this is a representation of how frequently those GC percentages are for these windows. So this gives you an idea of what the GC content of the genome is. Next on the right y-axis, we have mean base quality. So this is the mean base quality of the reads that map to these windows. So for this, it's considered to map to the window if the starting position of the read is in that window. The next part, which is the left-hand and y-axis, this is normalized coverage. So for this, a coverage of 1 is that it has the expected amount of coverage or number of reads for that at GC percentage. So this is based on how many, the frequency of that GC percentage in the actual genome. If you have lower than 1, this is a region that's underrepresented in the sequencing data. And then if you have it above 1, these are overrepresented regions. So you have more reads than you're expecting. So for the first one, we're going to look at humans. So this is GC content of 40%. And then for this, I am going to be showing full volume and then the miniaturized and then the 100 nanograms. So this is the highest input to compare the two of them to get against each other. So looking at the blue dots, which is the normalized GC coverage, you can see that the performance of the two library preparation techniques is very similar to each other. So the miniaturized has comparable performance to the full volume. The next up is Clostridium. So this is, a reminder, an 80-rich genome. So again, we have on the right we have miniaturized, and then on the left we have the full volume. And then looking at them here, I should add that these plots are representative of what the other libraries look like for each input and library type. So what you can see here is the performance is maybe even slightly better for the miniaturized compared to the full volume, but they are very comparable to each other. Next is E. coli. So this is like balanced genome, and again, similarly to the human, performance is similar. This time, the miniaturized is performing slightly worse than the full volume, but again, they are comparable. And then finally is the Rhodobacter. So this is the GC-rich sample that I was going to be showing. So again, performance is very similar between the two methods, where you can see a slight underrepresentation towards the low GC and high GC end of this genome. So next I'm going to have a look at the effect of DNA input on GC bias, again starting with human. So this is looking at the 4 different input types that we have for humans. So this is 100, which I'm showing on the bottom left, and then 20, which is bottom right. It's 6 nanograms, which is top right, and then 1.2 nanograms, which is top left. So for this, as you have a decrease in input, you can see the bias increases. So you look at the 1.2 nanograms, you can see that there is a clear underrepresentation of the higher GC content part of the genome for humans, whereas between the 6, the 20, and the 100, there isn't as much of a difference, and between 20 and 100, there is almost no difference. Next, looking at Clostridium. So to remind again, for this one, we do not have the 100 nanogram input. So it's just the 20, the 6, and the 1.2. So for this one, you can see a clear difference between the 1.2 and the others, and then a similar performance between the 6 and the 20 nanograms. Next is Rhodobacter. So this is the GC-rich one, and then again you can see the 1.2. You're getting more variation in the coverage comparatively to the 6 and the 20, and that the 6 and the 20 have very similar inputs to each other. Finally, I'm going to have a look at the effect of the chemistry on the GC bias. So again, we're starting with human, and for this one, we're going to show, so on the right, we have light. In the middle, we have 5x miniaturization. For this case, we're using the 1.2 nanograms input because this is the closest to light, so it's the most comparable. And then on the left, we have full volume. And then if you look at these, you can clearly see that light has underrepresentation of the low GC and high GC portions of the human genome, whereas the DNA prep and the miniaturized, those two have similar performance to each other. Again, the 1.2 has reduced performance comparatively to the higher input ones. So for Clostridium, which is the AT-rich, it's species from the mock community that I'm highlighting. So again, on the left is light, middle is 5x miniaturized, and then on the right it's full volume. So the miniaturized and the full volume have similar performance, whereas the light has a clear underrepresentation of the low GC portion of the genome. And then finally, for the Rhodobacter, so light has high overrepresentation, and then high overrepresentation of the low GC part of the genome, and then the low representation of the high GC. Whereas the miniaturized and the full volume have comparable performance to each other. So in terms of the conclusions from the validation experiment, so normalization may be required. So insert size normalization is still functional, but the yield is now variable. So you may need to normalize the DNA before you put it into library preparation if you want a consistent yield. The next is that DNA Prep is more tolerant than the original Nextera to high and low GC content. And next is that the miniaturized method is comparable to the full volume and gives similar performance. So we're not losing anything by miniaturizing. And then in terms of input range 20 to 100 nanograms gives the optimal all quality genome. However, if you wanted to have a low input, you could use the 6 nanograms, as the decrease in quality is not as severe, whereas we would not recommend 1.2 nanogram inputs, because the drop in GC by the increase in GC bias is a lot more severe in that, and also the yields from a 1.2 are actually make it quite difficult to pool and sequence. So finally, I'd just like to thank you for your attention, and then thank the following people for their help. So Paul and Kareem for helping lead this project, James Lipscomb, Anita and Harvey for helping develop the miniaturized DNA prep protocol, David and Yushuan for analyzing the data, Darren for the original development of Light and also providing the mock community, and then the rest of Genomics Pipelines for their help with the full volume for the full volume and the Light libraries that were sequenced. And thank you for your attention. I'm happy to take questions, and I'll pass over to Colin for the Q&A section.
Great, thank you, Thomas. As a reminder to our webinar participants, if you have a question, you can type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. I'll go ahead and get started with the questions. The first one we have here, does your team use a robotic arm to move from the Firefly to the PCR?
No. So for this one, was moved manually.
Got it. Did you test different input with the full volume DNA prep to check how well the yield normalization worked in standard conditions? And I'll add another one. Why do you think the yield normalization is not working with the miniaturized?
So for the first part of the question, no, we didn't test different inputs. So we were expecting in the normalization of the yield to perform as Illumina state. So over 100 nanograms, it will normalize. Below 100 nanograms, you'll see you won't get any normalization for yield anymore. So we didn't test for that. In terms of why the miniaturization is not working, so the normalization for yield is coming from that each transposase can only carry out one reaction, so insert a adapter sequence into your DNA. Once it's done that, it can't do it again. So once all of the transposases are occupied and have added an adapter sequence, and you can no longer create more library fragments. So my suspicion is why the miniaturization is causing the issue is that the number of DNA fragments does not necessarily scale at the same way as the DNA input reduction did. So my suspicion is that you're not getting the full occupancy of the transposase, and therefore you're getting in, as you increase the DNA input again, you are getting increased occupancy. So perhaps if you go over the 100 nanograms and tried some higher inputs in the miniaturization protocol, you might actually be getting the normalization that Illumina describes happening.
What were the major challenges that had to be overcome to develop your new method? What trade-offs, if any, did you make, and why?
Um, the sort of like the main ones that come with miniaturization and of any library prep method is normally particularly the mixing. So the smaller you go, the more difficult liquid handling becomes. So like mixing small volumes can be quite tricky, which is why we utilize both shaking and kit mixing in. So we had to do a lot of development time in terms of working out which one would be able to resuspend the beads efficiently enough without the risk of putting air bubbles into the bottom of the wells, which would then require you, because you want this to be an automated protocol, if you introduce air bubbles, you then have to take the plate and then centrifuge it down to remove the air bubbles, which means you need the human intervention. So we wanted to minimize that. So yeah, it's mainly just liquid handling challenges when you go to small volumes.
Is there a correlation between the genome size and the DNA prep kit capabilities to normalize samples?
I would say a no, although in general you do see genome, so like species to species variation in your yield and insert size. This is sort of like generally in terms of NGS libraries that you will see a certain species, so say wheat tends to have a different fragment and yield profile to human and samples for the same library type method.
Can you explain what the light sample is?
So for the validation experiment, this is using the same control human and the same mock arc. So this was so using the light library workflow that I described in the talk, we made 8 libraries from the human. So we made 6 human and 6 mock arc libraries, and then sequence them.
Got it. And how do you normalize beads to each input?
So for each input, we were adding the same. I'm assuming they're referring to the bead link transposase for this. So we use the same amount of bead link transposase for each one. So in theory, for the 20 and the 100, those should be within they're the 5x miniaturized equivalent of the 100 to the 500, which is what Illumina say. Between those ranges, you should get yield normalization and insert normalization. And so we didn't adjust the amount of beads for any of these. And then the other ones were just seeing effectively is low input viable, or for those we weren't necessarily expecting the normalization to work for them. It was to see, could we get good quality libraries out at the end, which for the 6, yes, for the 1.2, no, and I wouldn't recommend that one.
How long does the protocol take end to end?
So it takes between 3.5 to 4 hours. So this is dependent on the number of PCR cycles. For this, we had 5 PCR cycles. If you were to increase the number of PCR cycles to get more yield, then it would be closer to 4 hours. And I should also add that this is starting from the tagmentation reaction setup and then finishing on the final library. So that doesn't include the QC of the DNA or normalization of the DNA if you want to do it, and then the subsequent pooling and sequencing.
What was the main influencing factor in choosing Nextera-based library prep as opposed to mechanical shearing prep?
For this one, I mean, it is amenable to volume. So I believe there is an NEB method that is also amenable to miniaturization. The DNA Ultra Prep 2. I cannot off the top of my head remember if that's shearing based or whether it uses enzymatic. So the issue for mechanical shearing can be that if you start messing with the volumes, you can severely change the shearing patterns. Whereas for Nextera, particularly with the new technology, the bead-linked one, for the older one, changing DNA inputs can change the fragment size, resulting fragment size. For the new DNA Prep version of Nextera, you're getting more uniform insert sizes.
Got it. Again, if any viewers have questions, you can type it into the Q&A box in the control panel. We've got a few more minutes here. The next one, can the robot do the initial DNA QC and normalization, or is this all done by hand?
Yes. So I mean, we didn't obviously do it for this one because we were mostly focusing on the library construction method itself. But yes, normalization can be done quite happily with a robot. You transferred a fixed amount of your DNA sample with the varying concentrations into a plate, and then that plate can then have different volumes of a dilutant added to it by the dispensing in arm of a robot. So we use the Dragonfly eye to add different volumes, and then a fixed volume for the mosquito. And then obviously, you can then array, if you're just doing concentration, array the dye and the buffer, and then transfer your DNA into a different plate, and then take that to the plate reader. So these are all, very automatable. Similarly for the pooling afterwards.
Have you observed any evidence of well-to-well cross-contamination in your validation experiment?
So we've not observed any comms-com or row-to-row contamination for this. So I didn't mention them in the talk, but we also included 6 no-template controls in the library preparation. So these were distributed randomly throughout the plate, and none of these gave a signal or when we QC'd them. And then also we alternated between human and the mock community in terms of their well positions. And so you would have seen the human going into the mock and vice versa if there was contamination issue. So we didn't observe any of that in the sequencing data. The only caveat I'd add to that is that for this validation run, we only processed 3 columns of samples, so you need to test it out on a full column run to be absolutely certain.
Did you use the magnetic beads at the stock concentration or diluted?
We use them at the stock concentration. There is a dilution step so for the dual size selection in the Illumina as DNA Prep protocol, for the first spray, they're diluted in water, and for the second spray, they use neat. We miniaturized eyes, so we also diluted the beads for the first spray, but we reduced the volume of the beads and the water to dilute them consistently, as in 5x reduction in both. So it should be the same bead ratios as the full volume.
And what was the final volume for the miniaturized reaction?
So the final volume is 6 microliters, which is the 5x reduction in the elution, and then from that you probably get 4 microliters approximately for pooling for sequencing after you've taken off aliquot for QC.
Would there be an effect from genome size on coverage for low input libraries?
So we didn't observe this in our sequencing data. Obviously, for the human, and we did not sequence them at high, like we didn't have enough sequencing depth to get high coverage of the human and genomes. But in theory, for anything large genome size, the lower your input, the more likely you are to miss bits of the genome and therefore have reduction in coverage. There's still a utility to using low input miniaturized library preparation techniques. So if you're skim sequencing in your samples, for instance, you can still use a cheat, the fact that it's a cheap, so you can sequence more of them and skim sequence them. Their sort of like utility is more for samples where you have low input to begin with, or a small genome size.
Great. Well, that's all the time we've got for today. We'd like to thank Thomas Brabbs and our sponsor, SPT Labtech. If we didn't get to your question, we'll try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of the webinar or if you'd like to listen to it again, an archived version will be emailed to all attendees. And thank you for joining us. And thank you, Thomas Brabbs.
Thank you.